Assalamualaikum. The topic of our assignment is molecular farming with the title of tobacco, a highly efficient green bioreactor for production of therapeutic proteins. First of all, the history. The one who discovered the molecular farming is Dr. J. Christopher Hall. In the early 1990s, Canadian plant scientist Dr. J. Christopher Hall recognized the manufacturing and regulatory advantages of using transgenic tobacco plants to produce antibody drugs. So, what is the molecular farming? Molecular farming is the process of producing pharmaceutically important and commercially valuable proteins in plants. So, its goal is to provide a safe and low-cost method of mass producing recombinant pharmaceutical proteins. And molecular farming also has the potential to provide virtually unlimited quantities of recombinant protein for use of diagnostic and therapeutic tools in healthcare and the life science.
Tobacco is known as the white mouse of the plant world because it is genetically modified and has served as the primary vehicle for proof of concept work in recombinant protein production for the past 20 years. Tobacco is a leafy plant with a high biomass yield of up to 100 leaf biomass per hectare and a high level of soluble protein compared to many other models and plant species, all of which make it an attractive platform for protein production. Tobacco also provides a number of methods for expressing proteins of interest, including transient expression such as through agrobacterial or viral induction and stable nuclear or chloroplastic genome-based expression. Tobacco is not a food or crop, so the possibility of transgenic material contaminating food or the food chain is reduced. These properties have made tobacco a good option for the production of plant-based biopharmaceutical products. Indeed, the number of therapeutic proteins produced by transgenic tobacco plants is steadily increasing, with several tobacco-derived products progressing to human clinical trials.
How the molecular farming is done? Okay, in general, the first step, a DNA molecule carrying the genetic information for a pharmaceutical substance is introduced into the plant genome, and then the gene can be incorporated permanently or for a short period of time. The transformed plant acts as a bioreactor. The pharmaceutical active substance is extracted from the plant and then made into the formulated product. Production of pharmaceutical proteins in tobacco. Okay, first example is antibody. Okay, how? DNA that has been designed to make the antibodies is combined with the virus that can infect tobacco plant, and then the virus that infect the tobacco cells will produce a large amount of antibodies. So the antibodies is harvested from the leaf and purified from the tobacco plants to make a ZMAP. ZMAP is a cocktail of the three antibodies that can be quickly stick to the Ebola virus and clear it from the body. Second example is vaccine. Okay, first, synthesis. Okay, we synthesize the gene from the sequence of virus. Second is vacuum infiltration. We introduce the genetic material into the plant through the vacuum infiltration. Okay, number 3 is incubation. Okay, we let the plant incubate in the greenhouse for about 4 to 6 days to allow the protein expression and VLPs formation. Next is harvest. The plants are harvested in order to extract VLPs. Okay, followed by the purification. VLP is purified to obtain a clinical grade material. And the last one is Medicago vaccine, which is the immunological memory.
Next, cytokines is one of production of pharmaceutical proteins in tobacco. Cytokines are small proteins or glycoproteins produced by many different cell types. They are strong immunoregulators that regulate the intensity and duration of the immune response by stimulating or inhibiting the activation, proliferation, and or differentiation of various cells as well as their secretion of ABS or other cytokines. Cytokines act by binding to cytokine receptors expressed on the membranes of responsive target cells. Recombinant cytokines have been extensively studied as therapeutic agents for infectious and autoimmune diseases, as well as cancer. Although many recombinant cytokines are commercially available, their therapeutic use is prohibitively expensive. Furthermore, because most commercially available recombinant cytokines are E. coli derived, they lack glycosylation. Glycosylation is important for protein folding, stability, and function. As a conclusion, plants are proving their ability to be effective and efficient bioreactors for the production of pharmaceutically valuable recombinant proteins. A variety of plant species are being explored for use as green bioreactors, each with its own set of advantages and disadvantages. Tobacco provides a unique combination of biomass accumulation, ease of manipulation, protection against food chain contamination, and biocontainment not found in other plant species. With further technological advancement and product expansion, the future of much plant has the potential to become a positive force in improving the quality of life for all. That's all from us. Thank you.